And so now that we've discussed DNA, we're going to move into the actual central dogma and talk about gene expression. And so, as we know, if we want to pass genetic information from generation to generation, that happens through a process of DNA replication, or DNA making more DNA. But when we talk about gene expression, what we're really talking about is the central dogma, where information and the genetic instructions are passed from DNA to RNA during transcription and then ultimately translated into proteins during translation. Right? And so we take information from DNA, transcribe it into RNA, and then translate it into protein. And this process of DNA becoming something that we can see is what we typically know as gene expression. And so in order to talk about gene expression, we sort of have to start with explaining what a gene is. And so a gene is a basic nucleotide sequence, so it's the sequence of bases that codes for one or more functional products. And most genes encode proteins. And there's a specific part of a gene. So you can see a whole gene here. This is in the DNA, and this would be consisting of different nucleotide sequences. And the sequence of nucleotides that actually specifies that functional product or the amino acids that become the protein is known as the coding region. And so the coding region is from this line here to this line here, and that's the specific sequence that actually becomes translated into protein. It always begins with a specific start sequence, which is AUG, and ends with a specific stop sequence. And so the area between that start and stop codon is considered the coding region of a gene. And importantly, it's not just the coding region that matters in terms of genes. There are other parts of the gene that will effectively allow it to be transcribed and then also allow transcription of that gene to stop. And the sequence that's sort of upstream or before the coding region of a gene that helps transcription start or turn on is known as the promoter. And the promoter is a specific sequence that the enzyme that does transcription, RNA polymerase, can recognize and bind to start transcribing. The sequence of the promoter itself is not transcribed into an RNA or translated into a protein. So it only exists in the DNA instructions as a binding site for RNA polymerase. And then on the downstream side of the coding region, or after the coding region, there has to be a sequence within the DNA that tells RNA polymerase to stop transcribing that gene. And so this is a particular sequence in the DNA that RNA polymerase reaches after transcription and sees basically a stop signal, and the terminator will cause the RNA polymerase to dissociate from that DNA and for transcription to end. And as such, the terminator sequence or termination sequence is also not included in the mRNA or translated into protein. It specifically exists in the DNA as an instruction for RNA polymerase. And so you have the promoter region, the furthest upstream, then the coding region, which encodes your protein, and the terminator, the furthest downstream, which stops transcription. And transcription is just simply the synthesis of RNA under the direction of DNA. So it uses a DNA template to encode all different types of RNA. And one thing that's important for you to remember is that transcription doesn't just make mRNAs, which ultimately are the ones that get translated into protein, but also the tRNAs that are involved in translation, the rRNAs that make up the ribosome. Any type of RNA must be made via transcription under the instructions of the DNA. And the enzyme that does it, as I already alluded to on the previous slide, is known as polymerase. So RNA polymerase specifically. And that's just the enzyme that does transcription or RNA synthesis. And you can see here, this is a yeast RNA polymerase 2. And there are different polymerases based on which type of which type of RNA you're going to be synthesizing through transcription, polymerase IIs make the mRNAs. And so that's why I've included this structure here. And you can see that they have this sort of Pac-Man-like shape. Inside this empty space is where the actual nucleic acid DNA binds and where the mRNAs are made. And the rest of these are just multiple subunits that all have different jobs. And what's important to remember about polymerases is that they are made up of a bunch of different subunits. And because they are made of these different subunits, each subunit can have its own function, and RNA polymerase can do a lot of different things at the same time, which is important because of how transcription happens. And so transcription is broken into 3 steps. There's an initiation step, or a start. There's a step of elongation, where the mRNA or other types of RNA are actually made. And then there's a termination, or an end step. And so in order to initiate transcription, RNA polymerase first has to recognize and bind to that promoter region. It's the sequence in the DNA that it recognizes as the start point of transcription. And so RNA polymerase has a subunit that can recognize that and bind to the DNA. RNA polymerase has another subunit that can locally unwind the DNA. So normally DNA is in a double helical structure. It has those hydrogen bonds holding both strands together. But you need to separate that in order to have the template available to you to make RNA. And so RNA polymerase has one subunit that can actually effectively locally unwind the DNA and allow access to a template DNA strand. And then once the DNA is locally unwound there and open, there's another subunit as part of RNA polymerase that can actually add the complementary bases to generate that RNA strand. And so RNA polymerase binds, it unwinds the DNA, and then it starts adding bases. And it moves in a 5' to 3' direction down the DNA during the process of elongation, locally unwinding the DNA template, adding the complementary bases to create the mRNA strand, and then rewinding behind it as it goes. And so RNA polymerase only unwinds a little bit of the DNA at a time, and then rewinds it right after it's done transcribing that into mRNA. And during elongation, RNA polymerase continues traveling in a 5' to 3' direction until it reaches the termination site, or the terminator. And once RNA polymerase reaches that termination sequence, it sees that the gene has been fully transcribed and it can dissociate from the DNA. And since it was rewinding it as it moved, the DNA looks intact, right? It's double-stranded, it's all back together. And the RNA transcript can also dissociate from RNA polymerase and is now completely transcribed, right? And so you go through this process of initiating transcription, where RNA polymerase binds and unwinds and starts adding bases, all the way through elongation of making that mRNA strand in a 5' to 3' direction, and then to the termination where RNA polymerase dissociates from the DNA and you end up with a complete complete RNA transcript. And then after that transcript is made in the nucleus, it has to reach the ribosomes in the cytoplasm somehow. And there are a couple modifications that happen, particularly to mRNA transcripts within the nucleus, that help that mRNA exit the nucleus and enter the cytoplasm, as well as keep it stable so that it can ultimately be translated. The first of which is what's known as a poly-A tail, which is just a series of adenine nucleotides added to the end of the coding, to the end of the transcript. The other is what's known as a 5' cap that gets added to the basically 5' side of a transcript. And both of these contribute to the stability of the mRNA transcript, and they keep it stable even though it's a single-stranded molecule. And they additionally also help the mRNAs leave the nucleus through the nuclear pore complex so that they can start being translated. And in addition to the adding of the 5' cap and the 3' poly A tail to a transcript, there's also this process in eukaryotic transcription known as splicing, where that original transcript contains every complementary bases to every single nucleotide that existed in the DNA. But what ultimately gets encoded as protein is not this whole sequence, but only the sequences seen in purple. And so first and foremost, those orange sequences that won't become protein need to be cut out or spliced out because they don't contribute anything to the final protein. And so we have this kind of pre-mRNA that's generated from transcription, and then these orange portions, or the introns, are spliced out and removed within the nucleus so that only the important things that encode protein, or the exons, the ones that will ultimately code for the gene, are left, and then that spliced or cut piece of mRNA is what can leave the nucleus and be translated.